Hello everyone, my name is Subas and today I'm going to briefly present my work in the form of poster presentation. The title of my poster is Morphine potentiates glucocorticoid receptor translocation in neuronal cells. So a brief introduction is that HPA axis plays an important role in regulation of glucocorticoid receptor signaling, and as you can see on this slide, ACTH, which is here is the ACTH and here is beta endorphin. Both are derived through processing of pro-opiomelanocortin prohormone secreted from anterior pituitary under the stress conditions. ACTH is released in circulation, regulating the release of glucocorticoids from the adrenal glands, while the beta endorphins are the endogenous opioids. Glucocorticoid cause profound suppression of functional HPA axis activity, and the endogenous opioids activity acting primarily on the mu-opioid receptor inhibit the activity of HPA axis. Also, glucocorticoids regulate the MOR expression through binding to what we call a glucocorticoid response elements or GRE, but not in kappa or delta opioid receptor. Hence, we wanted to investigate whether GR activation results in its translocation to nucleus is modulated by morphine, which is a mu-opioid receptor ligand. So if you can see on this slide here is a glucocorticoid which binds to the glucocorticoid receptor, gets translocated into the nucleus, which is involved in transcriptional regulation. We wanted to find out if morphine modulates this nuclear translocation. So our methods, we used the neuroblastoma cell line neuro 2A, which was stably transfected with the either mu-opioid receptor or HSP70 or both. Cells were treated with morphine followed by corticosteroid, and after that we extracted the nuclear fraction and the cytosolic fraction. We also did one experiment where we used the immunocytochemistry using elixaflor 555 labeled secondary antibody. This is the first result here. So we wanted to see if HSP70 is being modulated by the morphine. We saw that HSP70 is increasing in a time-dependent manner, which was further confirmed by the qPCR. Then we also confirmed that these proteins together, like glucocorticoid receptor, HSP70, and mu-opioid receptor, all three of them are together interacting with each other in this case, in this scenario, which was confirmed by these immunoassays. We wanted to confirm whether the translocation of glucocorticoid receptor is being modulated by morphine or not. So we treated the G's with a different concentration of glucocorticoid in the presence or absence of morphine. So when the glucocorticoid was not present, there was no nuclear translocation. But other than that, whenever we used either low or high dose of glucocorticoid, it resulted in nuclear translocation. Beta-actin and RNA polymerase II are serving as a loading control for cytosolic protein and the nuclear protein respectively. When we quantified that, we saw that at lower concentration of glucocorticoid, morphine resulted in decreased translocation, while in all other concentration of glucocorticoid, morphine resulted in increased translocation of glucocorticoid into the nucleus. Here we are saying immunocytochemistry, so the red color is for the glucocorticoid receptor, the nucleus is stained with DAPI, and there is a merge. In this slide you can see that morphine by itself is not doing any translocation of glucocorticoid receptor to the nucleus, but when we used either 15 minutes or 30 minutes of treatment for glucocorticoid, either 100 nanogram or 300 nanogram per ml of glucocorticoid, morphine always potentiated the GR translocation. So if you can see here that whenever we treated with the glucocorticoid, there was still some amount of protein or receptor which is in the cytosol. When we treated with the morphine, all of them moved to the nucleus, so that potentiated that GR translocation to the nucleus. Also, we wanted to find out if this is a classical pathway or not, so we used the treatment of naltrexone, which is an inhibitor of mu-opioid receptor. So in cells which were which were stably expressing the mu-opioid receptor, we saw that the morphine resulted in increased translocation, while naltrexone resulted in decreased translocation. So naltrexone inhibited that. We saw the similar pattern in the cells overexpressing mu-opioid receptor as well as HSP70. So we confirmed with this experiment that probably HSP70 is playing some role in that pathway. So in the next experiment, wanted to see if HSP70 deletion can do something differently. So we knocked down the HSP70 using the shRNA. So in control cells, morphine increased the translocation, while in the knockdown of HSP70, that knockdown was completely inhibited. That means HSP70 is definitely playing a very crucial role in that. So based on our results, we conclude that morphine treatment time-dependently increased the HSP70 expression. Morphine treatment potentiated GR translocation neuronal cells. Overexpression of HSP70 facilitated GR nuclear translocation, while inhibiting HSP70 reversed the morphine-mediated potentiation. We also confirmed that morphine modulation of the nuclear translocation of the GR was through the classical opioid receptor pathway. With that, here is some references and acknowledgement, and with that we finish our presentation, and I would like to thank the committee to let me present this work. Thank you very much.